Welcome all in my lecture Modeling Server Swiss Model. You have learned different modeling server in this series. I am going to explain about advantages of modeling server Swiss Model. Swiss Model is a popular protein structure modeling server with several advantages that make it a valuable tool for researchers in the field of structural biology and related disciplines. First one is user friendly web interface. Swiss Model offers a user friendly web interface, making it accessible to researchers with varying levels of expertise. Users can easily input protein sequences and obtain 3D structure models with minimal technical hurdles. Second is large template database. Swiss Model maintains a comprehensive database of experimentally determined protein structures templates from the Protein Data Bank (PDB). This extensive template library allows for a broad range of proteins to be modeled, increasing the chances of finding suitable templates for modeling server for less studied proteins. Third one is automated template selection. The platform automatically selects suitable templates based on sequence similarities, simplifying the modeling process for users. It also provides multiple template options for users to choose from, allowing for flexibility in model generation. Fourth one is model quality assessment. Swiss Model provides tools and metrics for assessing the quality of the generated models. Users can evaluate models based on global model quality estimation and QMEAN scores, which helps in assessing the reliability of the predicted structures. Fifth one is a interactive visualization. The platform includes a built-in 3D molecular viewer that allows users to visualize and manipulate the generated protein models. This features helps researchers gain insights into the predicted structures and their suitability for specific research questions. Sixth one is model download and export. Users can easily download the generated model in various file formats such as PDB for further analysis using molecular modeling and visualization softwares. This flexibility allows for additional refinement or analysis as needed. Regular updates. Swiss Model is continually updated with new protein structure and improved modeling algorithms, ensuring that users have access to the latest resources and methods in the field of structural biology. Eighth one is a integration with other tools. Swiss Model is often integrated with other bioinformatics tools and resources, enabling users to perform a wide range of analysis such as functional annotation and ligand binding site prediction in conjunction with structure modeling. Ninth one is a community support. Swiss Model has a strong user community and researchers can find support, tutorials and documentation to assist them in effectively utilizing the platform for their research needs. 10th one is a free access. Swiss Model is freely accessible to the scientific community, making it an attractive option for researchers and institutions with limited budgets. Overall, Swiss Model is combination of a user-friendly interface, a vast template database, model quality assessment tools and regular updates make it a valuable resource for protein structure modeling, especially for those who may not have specialized expertise in computational biology or structural modeling. My colleague Jyotsna will explain the modeling server Swiss Model in detail.
Hello everyone. In this lecture, we will discuss about the modeling server repository. Homology modeling server are online computational tools that automate the process of predicting the three-dimensional structure of a protein based on its amino acid sequence and the known structure of homologous protein. that serve as a templates. This technique is particularly useful when experimental methods like X-ray crystallography or NMR spectroscopy are not feasible for determining the structure of a target protein. These servers make homology modeling accessible to researchers who may not have expertise in protein structure prediction or access to specialized software. In order to perform homology modeling, the two basic requirements are: first one is a sequence for which the X-ray crystallographic structure are not deposited in protein data bank and can serve as a query protein sequence, or target sequence and the sequence for which crystal structure are available on the database and have at least 30% or more than 30% similarity with the target sequence can serve as a template or model building. So, basically these two sequence, target and template sequence, are matched around 30% or more than 30%. They are known as homologous sequence. Anything below 30% identity will not be amenable for homology modeling, and we have to use other method of modeling such as ab initio modeling. The target sequence can be queried using different database accession code, that is UniProt accession code and ID, or GenBank ID or IPI, or RefSeq accession number, or directly with the protein amino acid sequence, that is the fragment thereof for a specific domain. Here are some well known homology modeling servers. First one is a Swiss Model. Swiss Model is a widely used homology modeling server that provides automated protein structure modeling services. It offers an intuitive web interfaces and allows user to input a target protein sequence, which is then used to search for a template structure in a protein structure database. The server then builds a model of the target protein based on identified templates. Second one is the Modeler. Modeler is a standalone software packages for comparative protein structure modeling. While it is not a web server, it is a powerful tool for researchers who want more control over the modeling process. Modeler can be downloaded and installed on a local computer. FIRE2. FIRE2 is a popular homology modeling server that provides protein structure prediction and analysis. It offers various modes for protein structure modeling, including intensive modeling and ab initio folding. User can submit a protein sequence and receive predicted structure. Next is iTesser. iTesser is an integrated platform that combines threading, ab initio modeling and structural refinement to predict protein structure. It also provides structure based function annotation and ligand binding site prediction. Rosetta. Rosetta is a powerful suite of software tool for protein structure prediction, refinement and design. While it is not a web server like other on this list, it is widely used in the field of structural biology. Researcher can access Rosetta software and perform homology modeling on their local system. RaptorX. RaptorX is a protein structure prediction server that uses deep learning and machine learning techniques to improve the accuracy of homology modeling. It provides three-dimensional model as well as predicted binding site and protein-protein interaction interfaces. EasyPred 3D. It is an automated homology modeling program. Alignments are obtained by combining weighing and screening the result several multiple alignment program. The final three-dimensional structure is built using the modeling package Modeler. FIRERISK. FIRERISK is a specialized server for modeling missense mutation in protein structure. It can be used to predict the structural effect of genetic variation. Bioinformatics Toolkit. The Bioinformatics Toolkit offer a variety of tools for computational biology, including homology modeling. It provides an interface to several modeling programs and servers. In this lecture, we will discuss in detail about the homology modeling server Swiss Model. Swiss Model is a widely used web-based homology modeling server and platform for protein structure prediction. It provides researchers with an automated and user-friendly way to generate three-dimensional models of protein based on their amino acid sequence and the known structure of homologous protein sequence, that is called templates. It is accessible via the Expressi Molecular Web Server, that is http://www.expressi.org. It is also accessible from DeepView Swiss PDB viewer program. DeepView provides manual control during homology modeling and tools for analyzing generated models. The purpose of these servers is to make protein modeling accessible to all life science researchers worldwide. Here are some key features and information about the Swiss model. Swiss model is having user-friendly interface. Swiss model offers an intuitive and user-friendly web interface that allows researchers to submit protein sequence and perform homology modeling without the need for extensive computational expertise. Large template library. Swiss model maintains an extensive database of experimentally determined protein structure from the protein data bank and uses these libraries to identify suitable templates for modeling. Automated modeling. The server automates the entire homology modeling process, including template selection, sequence alignment, model building and model evaluation. This automation streamlines the modeling process and provides quick result. Template selection. Swiss model selects the most appropriate templates for modeling based on sequence similarity and structural quality. User can also specify their preferred templates if needed. Model quality assessment. The server provides model quality assessment including Q-mean score, global model quality estimation score to help users gauge the reliability and accuracy of the generated model. Visualization. Swiss model offers an interactive 3D viewer that allows user to visualize and manipulate the predicted protein structure. This feature is useful for inspecting the details of the models. Additional tools. Swiss model offers additional tools and resources for protein structure analysis such as the Swiss PDB viewer and Swiss model repository for accessing pre-computed models. Customization. User can customize various aspects of the modeling process such as specifying the region of interest, using multiple templates and choosing modeling parameters. Submission and output. Researcher can submit their protein sequence through the web interface and receive the resulting 3D structural model in various file formats, including PDB file. The results also include detailed information on the modeling process and assessment score. Free access. Swiss model is freely accessible to researchers worldwide and does not require software installation or licensing fee. Integration with other resources. Swiss model is integrated with other bioinformatic resources such as UniProt database, allowing user to easily retrieve protein sequence and annotation. Swiss model is valuable resource for researcher in structural biology, bioinformatics and related fields. It enables them to generate accurate protein structure prediction quickly and efficiently. It is commonly used for a wide range of applications including drug discovery, protein engineering and understanding the structure function relationship of biological molecule. Swiss model is a structural bioinformatics web server dedicated to homology modeling of protein 3D structure. Homology modeling is currently the most accurate method to generate reliable 3-dimensional protein structure model and is routinely used in many practical applications. Homology or comparative modeling method make use of experimental protein structure, that is called templates, to build model for evolutionary related protein, that is target sequence. Today Swiss model consists of 3 tightly integrated components. The first one is the Swiss model pipeline. It is a suite of software tools and database for automated protein structure modeling. The Swiss model workspace, a web-based graphical user workbench. Next one is the Swiss model repository. It is a continuously updated database of homology models for a set of model organisms proteomes of high biomedical interest. The pipeline. Swiss model pipeline comprises the 4 main steps that are involved in building a homology model of a given protein structure. First is identification of structural template. First, BLAST is used to fetch homologous sequence, and if no suitable templates are found, HH-Blitz is used for detection of remotely related sequences. The templates are stored in the Swiss model template library, which is derived from PDB. Alignment of target sequence and template structure. Third one is the model building and energy minimization. Swiss model implements a rigid fragment assembly approach for modeling. Assessment of the model's quality using 3 independent methods: Q-mean, Anolia, Gromos. Then the workspace. The Swiss model workspace integrates programs and database required for protein structure modeling in a web-based workspace. Depending on the complexity of modeling tasks, different modes of usage can be applied, in which the user has different level of control over individual modeling steps: automated mode, alignment mode and project mode. A fully automated mode is used when a sufficiently high sequence identity between target and templates achieved, that is, sequence identity should be more than 50%. It allows for no human intervention at all. In this case, only the sequence or UniProt accession code of the protein is required as input. The alignment mode enables the user to input their own target template alignment from which the modeling procedure starts. The project mode is mode in more difficult cases when manual correction of target template alignment are needed to improve the quality of resulting model. In this mode, the input is a project file that can be generated by the DeepView, Swiss PDB viewer, visualization and structural analysis tool to allow the user to examine and manipulate the target template alignment in the structural context. In all 3 cases, the output is a PDB file with atom coordinates of the model or a DeepView project file. The 4 main steps of homology modeling may be repeated iteratively until satisfactory model is achieved. The Swiss model workspace is accessible via the Expressy web server, or it can be used as a part of program DeepView. The repository. The third component is repository. The Swiss model repository is a database of annotated 3D protein structure models generated by the Swiss model homology modeling pipeline. It is an up-to-date collection of annotated 3-dimensional protein models for a set of model organisms of high general interest. Swiss model repository is integrated with several external resources such as UniProt, Interpro, String Database and Nature PSI Protein Structure Initiative SBKB. New feature of Swiss model. The new development of the Swiss model expert system features. First is automated modeling of homo-oligomeric assemblies, modeling of essential metal ion and biologically relevant ligand in protein structure, local model reliability estimates based on the Q-mean local score function, mapping of UniProt feature to model. In above 4 options, option 1 and option 2 are available when using the automated mode of the Swiss model workspace, and option 3 is always provided when calculating a homology modeling using the Swiss model workspace, and 4th is available in Swiss model repository. Regular update of Swiss model ensures that target coverage is complete, that model are built using the most recent sequence and template structure database, and that improvement in the underlying modeling pipeline are fully utilized. It also allows user to assess the quality of the models using the latest Q-mean result. If a sequence has not been modeled, the user can build model interactively via the Swiss model workspace. Currently, the repositories contain 23,56,899 models from Swiss model for UniProt knowledge base target as well as 1,95,677 structure from PDB with mapping to UniProt knowledge base database. The accuracy and reliability of the Swiss model method. In the past, the accuracy, stability and reliability of the Swiss model server pipeline was validated by EVA-CM benchmark project. Currently, the Swiss model server pipeline is participating in the continuous automated model evaluation project, which continuously evaluate the accuracy and reliability of protein structure prediction services in a fully automated manner. Steps of homology modeling with Swiss model. Homology modeling with Swiss model involves several steps to predict three-dimensional structure of a protein based on its amino acid sequence and the known structure of related protein. Input protein sequence. Provide the amino acid sequence of the target protein in FASTA format. You can either paste the sequence or upload a text file containing the sequence. Select a template. Swiss model will search its database for suitable template structure. You can choose one or multiple template based on sequence similarity and availability. The tool will recommend templates, but you can also manually search for template if you have a specific one in mind. Next step is sequence alignment. Swiss model perform a sequence alignment between your target sequence and the selected template. This alignment helps determine how the target sequence relates to the template structure. Next is a model building. Using the template and the sequence alignment, Swiss model builds three-dimensional model of your query protein. It does this by transferring the structural information from the template to your target sequence. Next is model refinement. After the model building, the initial model may undergo a refinement step to improve its quality. This can involve energy minimization and optimization of the model's geometry. Model evaluation. Swiss model provides tools to assess the quality of the generated model. You can analyze various structural parameters such as stereochemistry, side chain orientation and class score. The platform may also provide a global model quality estimate score, which indicates reliability of the model. Visualization and analysis. You can visualize and analyze the generated model using the integrated molecular visualization tools available on the Swiss model platform, or by downloading the model for use with external molecular visualization software. Downloading of model. Once you are satisfied with the model, you can download it in a format suitable for further analysis or use in other software. Additional analysis. Depending on your research need, you may perform additional analysis such as protein ligand docking, molecular dynamic simulation or functional prediction using the generating model. Documentation and citation. If you use Swiss model, generated model in your research, it is important to cite the platform and any relevant template structure. Remember that quality and accuracy of your homology model depend on the quality of selected template and the alignment between your query sequence and the template sequence. Careful evaluation and refinement of the model are critical steps to ensure its reliability for further research and analysis. How to build a homology model with Swiss model. Here we provide a step-by-step example on how to build homology model for the our query protein superoxide dismutase, a protein from Drosophila melanogaster. This protein destroys free radical which are normally produced within the cell and are toxic to biological system. Its UniProt accession number is P61851. In its native form, this protein is a homodimer with a 2 metal binding site per subunit, one for copper and one for zinc. Access the Swiss model web server interface via swissmodel.xpc.org. This is the home page of Swiss model. Create an account if required or log in if you have an existing account. Then simply click on Start Modeling button. This will land you on the next page. Here you can either paste the protein sequence or provide the UniProt accession number of your query protein sequence in the input form. Alternatively, input formats can be selected using the opposite drop down menu. A project title is automatically suggested from the information retrieved from Swiss Model's own copy of the UniProt knowledge base annotation if a UniProt KB accession is used. To search for available template structure, click on the Search for Template button. When the template search is finished, the output page includes a main table showing the list of available templates ranked according to the expected quality of the resulted model. For each template, the following information is provided in the result section. A checkbox to select and visualize the template in the 3D panel, the SMTL ID of the template and a link to Swiss Model template library page associated to the SMTL entry, the protein name of the template, the coverage to the target sequence, darker shades of the blue refer to higher sequence identity, the global model quality estimation, the quaternary structure quality estimation, the target template sequence identity, the experimental method used to determine the structure and resolution if applicable, the oligomeric state of SMTL bio unit, the ligand present in the experimental structure, a clickable icon to expand the box with the description of the template. As you can see from the tables, there are multiple templates which cover the complete sequence and share a considerable sequence identity with our query sequence. Let us analyze them in more detail. By clicking on the checkbox, different template structure can be visualized and compared to structure superposition in 3D viewer. Let us rank them according to their coverage to the target sequence and let us select the 4 top ranking templates. We can observe that they all share a high sequence identity to the query sequence. The identity is more than 67%. As expected, their structure also look very similar as they superimpose very well. Some local differences are present at the level of loop region. Typically, due to the higher variability in terms of sequence and structure compared to the rest of the protein, loops are the region that are the most susceptible to modeling error. On the other hand, they are often involved in protein function, hence accurately modeling their structure is crucial important. To get an overview of the similarity between the selected template, simply click on the Sequence Similarity tab. An interactive chart shows the relationship between the templates in the sequence similarity space, and your target protein is represented as a filled red circle. Each template is displayed as a blue circle, where the thick blue R indicates target coverage. The distance between different template is proportional to the pairwise sequence similarity, that is, evolutionarily closely related templates will be clustered together. By clicking on a circle, you can look at the available information on the specific template. You can also select and visualize a group of similar templates by hovering over the cluster. The superposed structure of the selected templates will be instantaneously displayed in 3D, where you can inspect their structural differences. By looking at the alignment tab, you can notice that the sequence of the selected templates are very similar overall. Compared to the sequence of the target, they show some insertions and deletion as well as some amino acid substitution. By clicking on the cog icon, you can change the coloring scheme of the alignment by selecting one of the many available options. Let us select the cluster coloring scheme. As expected, the region that show most of the structural difference correspond to position in the alignment where amino acid substitution and indels are present. Even if these differences seems to have little or no impact on the overall structure of the protein, they can still play an important role for the function of the protein, that is ligand or substrate binding. If we go back to the template table, we can see that not all the selected templates have been solved in presence of the natural ligands, that is copper and zinc. In particular, only the top ranked template, after sorting by sequence identity, binds to the zinc ion, while the second base template structure contains both the zinc and the copper ion. After you have selected the templates, click the Build Model button to run the modeling job. For each model generated based on the selected template, the following information is provided: a file containing the model coordinates along with the relevant information on the modeling process, the oligomeric state of the model ligand, Q-mean model estimation result, the target template sequence alignment, the template name, the sequence identity to the target sequence coverage. By default, models are colored by model quality estimates assigned by Q-mean to highlight regions of the model which are well or poorly modeled. If several alternative models have been built for a target sequence, they can be interactively superposed and visualized. Local estimates of the model quality based on the Q-mean score functions are shown as a pre-recite plot and as a global score in relation to set of high resolution PDB structure, that is Z score. In the first plot below, a score is associated to each residue of the model reported on the x axis, reflecting the expected similarity to the native structure on y axis. Typically, residues showing a score below 0.6 are expected to be a low quality model. Different chains are shown in different colors. The second plot shows the quality of the model as normalized Q-mean score on y axis in comparison to the scores obtained for high resolution crystal structure. Higher values indicate that model is of comparable quality to experimental structure of the similar size. So, this is all about the modeling with Swiss model. Thank you.